Scientists spent decades collecting DNA samples from every corner of the planet. Indigenous tribes in the Amazon rainforest, nomadic communities in the Mongolian steppes, isolated island populations in the South Pacific, and remote villages in the Himalayas. They sequenced millions of genetic markers, built the largest database of human diversity ever created, and ran the computational analysis that would finally answer a question humanity has been asking for centuries. What they found shattered every assumption we had about race, ancestry, and what it actually means to be human. The results were so unexpected that some researchers initially thought their equipment was broken. The data showed patterns nobody predicted, connections nobody saw coming, and revelations that made headlines across every major scientific journal in the world. Stick around, because by the end of this video, how you view human history, your own lineage, and the concept of race itself will be completely transformed. Before we dive into the data, make sure to hit that subscribe button and ring the notification bell. We break down groundbreaking scientific discoveries like this every single week. The Global Mapping Project. For several decades, an international coalition of geneticists, evolutionary biologists, and anthropologists undertook one of the most ambitious scientific endeavors in modern history. Teams of researchers traveled to the most isolated regions of planet Earth, seeking to collect biological samples from populations that had remained geographically distinct for thousands of years. They documented genetic markers from indigenous communities deep within the South American rainforests, nomadic herdsmen moving across the vast Mongolian steppes, isolated island societies in the remote South Pacific, and high-altitude mountain villages tucked away in the Himalayas. The primary objective of this massive endeavor, most prominently executed under initiatives like the Human Genome Diversity Project initialized in the early 1990s, was to construct an exhaustive global archive of human variation. Scientists sought to read, map, and analyze millions of genetic sequences to definitively resolve questions regarding human ancestry, population divergence, and the biological validity of classical racial classifications. At the time of its inception, the project generated intense ethical debate. Detractors expressed deep concern over the potential commercialization of genetic material from vulnerable indigenous groups, while advocates maintained that capturing this data was essential prior to the inevitable blending caused by modern international travel and rapid globalization. Regardless of the surrounding controversy, the scientific community agreed on a central premise: analyzing petabytes of genomic data across global populations would fundamentally alter our understanding of human history. The study sampled individuals across dozens of distinct geographic regions, spanning Africa, Europe, the Middle East, Central Asia, East Asia, Oceania, and the Americas. Advanced high-throughput sequencing technology allowed researchers to decode billions of base pairs of deoxyribonucleic acid, DNA, in parallel. When the initial computational runs concluded, the data triggered immediate astonishment across research facilities. The empirical results contradicted centuries of accepted scientific dogma, social consensus, and biological assumptions. The biological markers showed patterns that defied standard predictive models, demonstrating that traditional concepts of race lacked a true genomic foundation. The fallacy of biological race. When analyzing physical appearance, human beings display striking exterior differences. Features such as skin pigmentation, cranial structure, hair texture, and eye shape vary significantly across different latitudes. However, when computational biology evaluates the underlying genetic architecture, these superficial variations account for an extraordinarily minor fraction of the overall human genome. every human being on Earth shares approximately 99.9% of their genetic code with any randomly selected individual anywhere else on the planet. The entirety of human variation, all physical traits, disease predispositions, and physiological adaptations resides within the remaining 0.1% of our DNA. Crucially, the genetic analysis demonstrated that the 0.1% of variable DNA does not cluster into distinct, non-overlapping racial groups. Instead, genetic variation exists along a continuous spectrum, known scientifically as a cline. Traditional boundaries between geographic populations dissolve under rigorous genetic screening. An individual from Northern Europe may share far more specific genetic markers with a person from East Africa than two individuals living in adjacent geographic regions within Africa share with each other. Furthermore, populations that appear visually similar due to equivalent environmental adaptations often share entirely different genetic mutations. For instance, dark skin pigmentation in southern India and among indigenous Australians evolved through entirely distinct molecular pathways, despite yielding comparable visual outcomes. This phenomenon, known as convergent evolution, occurs when unrelated populations independently adapt to similar environmental pressures, such as high ultraviolet radiation, without sharing immediate lineage. The physical traits historically used to define racial categories, primarily skin color, facial morphology, and hair type, are governed by a tiny subset of genes within that 0.1% variable fraction. These visual features represent superficial adaptations to localized climates rather than fundamental divisions in human biology. The African origin and the founder effect. The most fundamental discovery revealed by global genomic mapping is that the vast majority of all human genetic diversity is contained within the African continent. Because modern Homo sapiens originated in Africa approximately 300,000 years ago, ancestral populations lived, adapted, and diversified within Africa for hundreds of thousands of years before small groups expanded to populate other continents. Because of this long evolutionary history, two individuals from different regions or even neighboring communities within Africa can exhibit greater overall genetic variance between each other than a randomly selected African individual exhibits when compared to a non-African individual from Europe, Asia, or the Americas. The genetic structure of non-African populations can be understood through a population genetics concept known as the founder effect. Approximately 70,000 years ago, a relatively small cohort of individuals migrated out of Africa into the Middle East, eventually spreading across Eurasia, Oceania, and the Americas. This migrating population carried only a tiny sampling of the total genetic diversity present in the original African source population. As subgroups continued to travel further eastward and northward, each subsequent migration event represented another bottleneck, carrying a progressively smaller subset of genetic markers. To conceptualize this process, imagine a container containing 100 uniquely colored marbles representing the full spectrum of human genetic variation developed in ancestral Africa. If a researcher selects a random handful of 15 marbles from that container to establish a second collection, that new collection will naturally lack many of the original variants. Even if those 15 marbles multiply and spread across millions of new individuals, they can never spontaneously recreate the lost 85 colors of original diversity. Consequently, genetic diversity systematically decreases the further a population is located from the African point of origin along ancestral migration routes. European populations exhibit less overall genetic variance than Middle Eastern populations. East Asian populations exhibit less variance than Central Asian groups, and Native American populations having crossed the Beringia land bridge during late migration waves, display the least overall genetic variance of any continental group. If you're finding this deep dive into genetic science fascinating, drop a like on the video. It really helps the algorithm push these educational breakdowns to more curious minds like yours. And leave a comment down below. Did you realize that all human physical differences come down to less than 0.1% of our DNA? Let's keep the discussion going. The myth of population purity. Beyond disproving the biological reality of discrete races, genomic research thoroughly dismantled the notion of ancestral purity. Ancient DNA extraction, aDNA, which allows scientists to sequence genetic samples directly from ancient skeletal remains, reveals that human history is defined by constant migration, overlap, and genetic admixture. European lineages. Sequencing of prehistoric remains proves that modern European populations are not direct lineal descendants of original Ice Age hunter-gatherers. Instead, they represent a three-way blend of Mesolithic hunter-gatherers, early Neolithic farmers who expanded from Anatolia roughly 9,000 years ago, and Yamnaya pastoralists who migrated from the Eurasian steppes during the early Bronze Age roughly 5,000 years ago. East Asian lineages. The Han Chinese population, frequently mischaracterized as a homogeneous group, exhibits significant genetic divergence between northern and southern regions, reflecting thousands of years of population movement, imperial unification, and internal migration along major river basins. Americas and Oceania. Genomic mapping of populations throughout the Americas reveals complex ancestral layers, including ancient genetic signals linking certain Amazonian tribes to early populations in Australasia, confirming multiple complex waves of oceanic and terrestrial migration. These findings show that isolated, unmixed ancestral populations do not exist in human history. Human lineages do not branch out like isolated trees. Rather, they form an interwoven network where distinct strands continually merge over time. Environmental selectivity versus biological race. If traditional racial categories do not align with genetic realities, it raises an important question. Why do certain hereditary medical conditions cluster within specific geographic populations? Diseases historically cited as evidence of fundamental biological differences, such as sickle cell anemia in West African populations, Tay-Sachs disease in Ashkenazi Jewish communities, or cystic fibrosis in Northern European groups, are not products of racial categories. They are the result of localized natural selection responding to specific environmental pressures or historical population bottlenecks. Carrying a single copy of the sickle cell mutation provides significant protection against mortality from severe malaria infection caused by Plasmodium falciparum. In regions where malaria was historically endemic, individuals carrying this genetic variant were substantially more likely to survive and reproduce. Consequently, the gene frequency increased in malarious areas, including parts of West Africa, the Mediterranean basin, the Arabian Peninsula, and southern India. An individual from a historically malarious region of Greece or Italy may carry the protective sickle cell trait, whereas an individual from an unexposed region of southern Africa may not. The distribution of the gene follows the geographical presence of the parasite, not arbitrary racial boundaries. Utilizing race as a clinical proxy for diagnostic risk is biologically inaccurate compared to direct genomic screening. Common ancestry and mathematical convergence. Genomic research combined with statistical genealogy yields astonishing conclusions regarding how closely all living humans are related. Because every individual has two biological parents, 4 grandparents, 8 great-grandparents, and so on. The number of direct ancestors required by an individual's family tree grows exponentially as one moves back through generations. Ancestors per generation equals 2 to the power of n. N equals number of generations past. If one tracks this progression backward by just 30 to 40 generations, roughly 1,000 years, the mathematical number of ancestral nodes exceeds the total number of human beings alive on Earth at that time. This mathematical reality indicates that individual family lines constantly cross, overlap, and merge. Genetic models confirm 2 profound genealogical benchmarks. The most recent common ancestor, MRCA. Computer simulations and genomic tracking demonstrate that the most recent common ancestor of all 8 billion living humans lived astonishingly recently, approximately 2,000 to 3,000 years ago. The genetic identical ancestors. Point. Moving back further to approximately 5,000 to 7,000 BCE, humanity reaches a point where every person alive at that time who left living descendants is a direct ancestor of every single human being alive today. Whether considering a farmer in ancient East Asia, a hunter-gatherer in sub-Saharan Africa, or an early agriculturalist in the Fertile Crescent, if their lineage survived, they occupy an identical position on the family tree of every modern human. When scientists decoded the DNA of humanity, they didn't discover evidence to divide us. They found proof of our undeniable connection. Beneath the surface-level adaptations that helped our ancestors survive in different environments, we are all part of one continuous family tree. If this video expanded your perspective, share it with someone who loves science. Don't forget to hit that subscribe button, hit the notification bell, and check out our playlist on the screen right now for more fascinating deep dives into the science shaping our world. Thanks for watching, and we'll see you in the next episode.